A very brief overview of what we know so far about Martian biomarkers and what we have learned using robotic missions. I'm focusing on three specific missions: Curiosity, Perseverance, and the Mars Sample Return architecture. But of course, these lessons have implications on other future missions and also the interpretation of past missions that went to Mars before. As a brief introduction, when we're talking about looking for biomarkers, it's basically that we're looking for things that point to life. And for that, we need to define previously what is life. What is it that we define as life? So we can choose one definition or another. I'm going to use here a NASA's-like definition. Life is a self-sustained chemical system capable of Darwinian evolution. Now, whatever life is, the only system where we have seen life is on Earth. So we have to see what made Earth be so interesting for life to appear, and what is it that is common to all known life forms. And what is common is that all the forms that we know of use organic chemistry, and this chemistry is based on the use of certain atoms like carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphor, among others. And all these atoms, which were available in our planet and are available in other planets in the universe, have recombined at some moment because of the existence of electric discharges or other sources of energy. They have been broken apart from gaseous or liquid precursors, and they have formed these basic fundamental building blocks of life. So, for instance, you could start with a prebiotic atmosphere with nitrogen or CO2 or water vapor, hydrogen. And if you apply certain energy sources, those molecules break apart and they recombine. And we know from prebiotic chemistry that you can form these very interesting building blocks of life. Once these are formed, they are not indicators of life, but they are precursors to life. So they can combine and they can form some long polymers with very specific functions, like phospholipids, which actually create the lipid membranes, or they can create DNA and RNA polymers that are the ones that carry information for all the life forms that we know of. And they can also create peptides, and those are the ones responsible for metabolism. So this is a kind of a very simplistic definition of life and how we understand it on Earth. And whatever happened on Earth, it finally led to the formation of one cell or multiple cells, who knows, maybe there were multiple initial origins of life. And then life evolved because of this Darwinian evolution that we talked about. So bearing this definition in mind, what happens when these processes evolve in other planetary environments? So if they evolve, then they may leave some imprint that we can look for. And that's what we define as a biomarker or biosignature. The biosignatures can have different nature. I'm going to give a few examples. The first one would be at atomic or elementary level. And that is that, for instance, you can have alteration of the presence of certain critical elements or the isotopic concentrations of elements. Isotopes, for instance, in the case of carbon, you've heard about carbon-14 and then carbon-13 and 12. Those are stable forms of carbon. And we will see later on how life favors one isotope over the other. So over time, if there is a living system using carbon, there is a tendency to concentrate one of these isotopes over the others. Now, another way of looking for imprints of life would be at a molecular level. So let's think we're looking at Earth. Sorry for the small size of this graph, but what we're looking at, we're showing here is the concentration of oxygen over nitrogen here over time, measured in the atmosphere of Earth. And here we're seeing the concentration of CO2. And as you can see, there is an oscillatory behavior. And this is basically a demonstration of all the photochemistry, all the chlorophyll activity of all the biomass on Earth. So we are seeing the planet as a whole breathing, capturing CO2 and releasing oxygen. Ideally, if we could look at gaseous molecules that are associated with metabolism in other exoplanets, we would be super excited and say, oh, maybe that's a potential biomarker. Now, exoplanets are super distant, so these faint signatures are very difficult to be detected. And obviously, there are many processes that can also produce these and are not associated with life. So one has to be very cautious. Now, another biomarker is at the level that I was talking about before, is that when we are targeting biomolecules with very specific functionalities, like the ones we discussed about, like peptides or lipids or DNA, those are kind of very exciting and very promising biomarkers. But the problem with these molecules is that they can degrade rapidly once the cells die. The next kind of confirmation that you may have would be a mineral footprint, like if you see precipitated minerals that only appear because there were some organisms eating the rocks, or if you see certain redox alteration patterns in the minerals, or much better, if you see something that looks like a biomorph, a fossil, right? Now, those processes also, let's say this could be a fossil, you could have things that look like life, but there's some of these footprints can also be generated by things that are not associated with life. And this applies to all of the potential biomarkers that I said, except for proteins, DNA, and RNA. Those are very specific for living organisms. So looking for biomarkers is definitely a challenge, because when we are looking for ancient life, you need something that preserves them. And then if you're looking for actual life, especially in environments like Mars, you have to be aware that if there is life, there's going to be in very small concentrations. So you need to have extremely highly sensitive detectors or instruments. So to add complexity to this, for either one of these analyses, you need to be aware that since on Earth there is life everywhere, whenever we look for life elsewhere, we may bring it with us. So we have to make sure we avoid any cross-contamination and we don't detect ourselves when looking for life elsewhere. So why are we looking for life beyond Earth? I'm going to give one example. It's kind of a very recent result that was published this year in Nature Astronomy. And it's an example of a sample return mission. But in this one, it's not from Mars. It's a sample acquired by the OSIRIS-REx mission from Bennu asteroid. And this asteroid was formed a long, long time ago, and like 4.5 billion years ago. And it was broken apart a long time ago, and it's been traveling through space until the moment when the OSIRIS-REx mission went and captured some of its material and brought it back to Earth. And if you analyze those samples in ultra clean conditions, it's very exciting to discover that you have the fundamental ingredients of life, or some of them, in these elements, in these rocks that are floating in space. 14 out of the 20 amino acids that make up proteins on Earth have been found there. Isn't this exciting? So this basically tells you that the fundamental ingredients for life may be formed elsewhere, maybe in the solar system, and why not, they may also form in other environments like Mars. The reason why we're looking at Mars as our prime target for the search for life is because the primitive Earth and the primitive Mars looked like very similarly. This is an example of how both planets looked like 4 to 3 billion years ago. When we think there was a water cycle, there were volcanoes, there were atmospheric discharges. So both planets had essentially the same geophysical processes. Now, what happened on Earth is that eventually, if this is the clock of life, if the planet was formed here, and this would be in a scale of billion years ago, eventually life appeared. We don't really have a record of the very origin of life because those rocks have disappeared. But we know that eventually phototrophic bacteria started to proliferate, and then there were cyanobacteria, and they were releasing oxygen to the atmosphere. And then all life forms evolved on Earth and evolved together with the planet. The reason we are going to Mars is because Mars kind of remained almost frozen in the state that it was when life appeared on Earth. And the question that we're going to pose to the rocks of Mars is, do you have any imprint of what happened there? Is there any prebiotic chemistry, or is there even any life that has been preserved and has been surviving there? Now, prebiotic Earth and maybe prebiotic Mars also look like this. This is just an artistic impression of how it may have looked like. And we know from experiments and from measurements in environments, in remote environments on Earth, that starting from very neutral atmospheres, you can produce the synthesis of organic elements. You can also have it in hydrothermal environments. You could also have, as we said before, organics coming from extraterrestrial delivery, like the ones from Bennu. And also there are certain materials and certain minerals where you can have chemistry that is induced in interfaces, especially where you have drying and wetting processes. So whichever was the geological or geophysical context of the emergence of life on Earth, something similar may have happened on Mars too. And that is why we have put our eyes in this planet. This is a topographic representation of the present day Mars. And you can see here labeled the missions that have landed already in the planet. So we are kind of having very local views of how Mars looks like and how it may have looked like in the past. All these missions are complemented with orbital measurements from remote sensing instruments. So we kind of interpret the minerals and the history. And we know for a fact that there was an ocean here, and then there were rivers, and there were even glaciers. So one of the missions that was launched to Mars to study the habitability of the planet, one of them was Curiosity, which landed here near the equator. And the other one is Mars 2020, or Perseverance, that landed in Jezero Crater. And this is a view of these twins. As I said, Curiosity, also called the Mars Science Laboratory, it's a huge platform that was developed to carry along a number of instruments or payloads, whose main goal was to characterize Gale Crater and evaluate if it was habitable in the past. Now, after the huge success of this mission, there was a new mission that was developed, and this one is Mars 2020, or Perseverance, and it landed in 2021. And it's exploring a different region of the planet, and it has another goal in addition of characterizing this environment. And the goal is to capture a collection of samples to be investigated on Earth. We're going to see first a brief summary of the discoveries of Curiosity. These are very nice postcards of what the landing site of Curiosity looks like. And from that, we're going to move into the discoveries of Perseverance. Well, after a few years of study of the Gale Crater region, Curiosity discovered that this was the way it may have looked in the past. What now is a crater that is empty, in the past was apparently filled with water. There was a river that was bringing materials, and then through different epochs of this region, we have seen salts that precipitated, and there are different layers as the rover now is climbing up the mount with all these different histories or strategic risks that represent different epochs of the planet. But when you analyze the composition at small scale, what the rover concluded using the measurements from Curiosity, it was concluded that this environment was habitable. There was water, there are organics that have been preserved, there was nitrogen as nitrates, there are phosphates, there is iron and sulfur available, and there are different redox states in the minerals. So in principle, this environment could have hosted life. There are other indicators that have been provided by Curiosity that could be interpreted as potential biomarkers. I'm going to give a few examples. One of them are atmospheric measurements. Curiosity has the possibility to measure the concentration in the air of methane, and this is an example of the measurements provided by SAM over different periods of one Martian year. And clearly, methane was detected in very tiny amounts. Also, it can detect the presence of oxygen, and surprisingly, it detected variations in the concentration of oxygen. Methane has been proposed sometimes as a potential biomarker because on Earth it's mostly produced by microorganisms, but there are other processes that can also produce it. So these are weak biomarkers, but at least they are potentially interesting, and they are indicative of anomalous dynamics in the atmosphere. The most powerful biomarkers today are the discovery of organic molecules in the samples that have been analyzed by Curiosity. I'm showing here an example of the shipbed mudstones. So these mudstones and the sandstones here in this region were analyzed extensively by Curiosity. The way it's done is that first, once that a region of interest has been discovered, then the materials are drilled and then they are put inside the rover instruments and they are analyzed. And this is a summary of some of the small organic molecules that have been discovered through time. As you can see, over more than 10 years, the samples have been analyzed. And even reanalyzing some of the initial samples lately, just this year, it has been published that long chains of carbon and hydrogen have been detected and preserved. They have been preserved on Mars. Why is this potentially interesting for us? Well, the discovery of these long chains of hydrocarbons may be related to the presence of degraded fatty acids. These are the longest chains that have been discovered so far on Mars, and their presence is still a weak indicator of life, but it's another potential added evidence for life in the past. Now I'm going to move to another indicator of the ones I introduced at the beginning. Remember that we discussed the idea of isotopes. I'm going to give an example of how it works on Earth. So please follow this explanation carefully. From chemistry, we know that you can have three stable forms of carbon. And on Earth, generally, carbon-12 and carbon-13 are in a ratio where we have a lot of carbon-12, like almost 98.9%. So let's imagine you have this in the air, molecules of CO2. I'm marking in yellow the ones that have an isotope of 13 of carbon, and then in green the ones that have 12. What happens is that if you have a process like photosynthesis, plants are going to, over time, tend to select the molecules that are composed of carbon-12. I'm exaggerating here how it happens, but over time you're enriching the organic matter that photosynthetic plants are fixing in carbon-12. So in other words, you are depleting this organic matter in carbon-13. Whereas if you have other abiotic processes like carbonate precipitation, then you don't have this strong selection. So over time, when you analyze something where the carbon has been processed through a biological process, if you have the biological process, you would have kind of this anomalous concentration. And if you don't have it, then you have this other ratio. This is an oversimplification because there are many processes that can alter these isotopic ratios. But this is a way to illustrate why we're looking for isotopic ratios alterations on Mars. I'm just giving here a summary of recent references of analysis in situ, all of them produced with Curiosity rover. And I'm going through examples of analysis of carbon-13 isotopic ratio. And as you see, some of the samples that have been analyzed show a negative, a depletion in carbon-13, which is kind of comparable to the one we see on Earth in organic matter. They have also analyzed the ratio of sulfur, and some of these ratios may be compatible with abiotic photolysis and definitely point to sulfur redox cycling. The oxygen isotopic ratios have also been investigated, and particularly the carbon-13 in methane released from pyrolysis of rock samples has shown very low negative values. Actually, it also shows positive values, but this kind of points to something that is compatible with the observations on Earth of biogenic methane. Again, these footprints are very weak and they cannot be interpreted on their own. They need to have complementary measurements to be called a biomarker. I'm also including, for completeness, other analysis of isotopic ratios, because these elements are actually used to interpret what is the evolution of the atmosphere of the planet. And they clearly indicate that there has been a lot going on in this time. So we know that the climate in the planet has changed, and whatever happened in the surface, if there was life there, it has had to adapt and evolve under different environmental conditions. So that was a brief overview of the main discoveries of Curiosity rover. And now we're moving to another site on Mars. We're going to Jezero Crater. And here we have in the front Perseverance rover. And we also have the drone that was flying along with it at the beginning of the mission. And what you can see in these small images is a video of how the rover captures samples on Mars. On the left, you have the regolith sampling, and on the right, you have the drill of a solid rock that is going to be put inside a tube and sealed. And why is this mission doing this? Well, as I said, Perseverance not only has payloads or instruments to do analytical studies in situ, but also it has the possibility to prepare a collection of unique samples that may be brought back to Earth in the future. The samples that have been acquired so far are two collections. One of the collection is within the rover, and the other collection was dropped on the floor of the crater and is called the backup collection. The purpose of this mission is to prepare a collection that is very diverse, and it covers very different periods of the history of the evolution of Mars. And by analyzing those samples with high quality measurements on Earth, we may infer things about the evolution of Mars, not just about the presence of life. Now, again, based on remote sensing observations and on in situ observations, this is what we have interpreted that Jezero Crater looked like in the past. When life appeared on Earth, Jezero was filled in with water. There was a lake, there was a river that was pouring a lot of water and material here, and then eventually there was so much water that part of it was just running out through this outlet. Over time, as I said before, the atmosphere changed, the climate changed, and then the water was lost. And what happened is that in this region, particularly, we can see now a dry surface with the remnants of a delta that was formed when the planet had water and a very intense liquid water cycle. What we are showing here is the route that the rover has followed from the beginning of its operation on the surface of Mars, and every red point corresponds to a place where a sample has been acquired. Before we introduce the samples and the potential biomarkers, I want to go back in time and try to interpret the geological context of these samples. This is a remote sensing observation of Jezero Crater, and you can see marked in colors different regions of interest. You can see regions where olivine has been detected and carbonates, and you can also see the crater rim and the river with a meander, and then the crater floor. Now, every region was produced in a different time in the history of Mars. First, we had the impact that was produced a long time ago because of an impactor that crashed on the surface and then produced this crater rim region with ejecta. So the rocks that we have on the top will be very hard and will be very old. Eventually, there was water on Mars, and there were processes that were producing olivine, and then there were processes that were fixing CO2 and producing carbonates. Over time, this evolved until we had this delta forming on the left, on the west side, but also there is one on the north that I didn't talk about. Eventually, this was covered with water. Finally, the water disappeared, and there was some kind of igneous process later on that covered the base of the crater with igneous material. So if we are able to sample these different units, what we're going to have is a time capsule with rocks that have a registry of different moments of the history of Mars. This could be kind of a very oversimplified view of the time evolution of the two planets, Earth, where you have the late heavy bombardment affecting both planets. And then we have here the oldest rock record on Earth and the time when life appears on Earth. In parallel, Mars has a similar but different history. And we believe that the Jezero impact was formed over here. The carbonates and olivine region were formed over here, the river and the lake. And then the period where everything was covered with the igneous material happened much later. Now, it's super exciting to think that the rover can actually capture rocks that were produced in these different periods, and we can investigate what happened before, during, and after the emergence of life on Earth. This is a map of the way that the campaigns have been planned so far. As I said before, there was the landing took place here, and the very beginning we were sampling in the crater floor. Then we went towards the delta front and we dropped a collection of samples here. This is the backup collection. Then the rover continued up the delta and started exploring these materials that are formed by very fine grain materials. They're sediments. And now we have passed the margin and we are exploring the crater rim blocky unit. This is one example of how it looks like now on the surface of Jezero Crater. It's super arid, and this material has been demonstrated later on to be indeed formed by igneous processes. And this is the way that it has been confirmed from the surface that there was a delta that was pouring water into a lake. And the things that we now see from the surface are this kind of delta remnants where you have some inclined foresets and you have the fluvial deposits, you have huge boulders that are accumulated on the top, and you have very fine material that has settled and in the bottom sets. And the exciting thing about this mission is that we have sampled here and up there. Now the backup sample collection is what you're looking at here. Eventually, before going up the delta, we dropped a small subset of samples. In case the rover fails in the future, we know for sure that this sample suite has enough content to be scientifically interesting. However, the collection that is within the rover has, of course, much more added value because it has a diversity of rocks, and then it's covering a wider period of time and different geological processes. However, already within the Jezero Crater floor, the instruments on board Perseverance were able to confirm the presence of organics in the rocks. And not only that, but these organics that are not that old, they were basically concentrated within regions that are produced by interaction of water. So that's kind of interesting because it points to a place where water may have concentrated organics. Now, as we climbed the delta, we found new rocks. Some of them had different visual potential biomarkers. I discussed before that a potential biomarker could be the formation of redox gradients or patterns. And this is one of those examples. You can see what was named the poppy seeds and the leopard spots. This could be a leopard spot, and the poppy seeds could be this one. And the instruments on board Perseverance have analyzed the concentration of organics and the presence of phosphor, iron across these patterns. And it's very interesting because these patterns form on Earth when you have microorganisms eating on the rocks. Of course, they can also be formed by abiotic processes, but again, you're adding one key element more into this view of the existence of life and how biomarkers may be preserved. So altogether, this gives you a view of what in situ robotic missions can do. You're always limited by somehow serendipity. You're limited by the instrument's capabilities, the duration of the mission. And of course, if we had the possibility to analyze samples on Earth, where we have our equipments, where we have laboratories, where you could have different scientific teams investigating these same samples, then the amount of discoveries that we would do would be much more confident and coherent, right? So the analysis of the Martian samples on Earth, or maybe even like the future human exploration of Mars, is important for us to understand the evolution of the planet, but also to have a view of what happened on a habitable planet when life was appearing on Earth. We may not find life, but we may find the prebiotic precursors. When we do this, whenever we put our hands on Martian rocks, we have to be aware that we're putting in contact two potentially habitable environments, and one of them is full of life, that's Earth. So we have to make sure that we create barriers, physical and chemical, to prevent contamination from the samples. Otherwise, it's very easy that any of these small bacteria or small, even the small molecules that are present in every instrument on Earth, in every product in the lab, even in the air, they can be transferred to the samples that we are analyzing. So it doesn't matter if we are analyzing them on Mars or if we are analyzing them back on Earth, we need to make sure that the necessary planetary protection measurements are put in place. Let's see another kind of biomarker, one that is more intuitive to us. Long time ago, there was a meteorite that came from Mars, the Allan Hills meteorite, that was analyzed on Earth. Meteorites are Martian samples, but they are not pristine. They have been exposed to the transfer through space. They have been contaminated as they went through the atmosphere. And obviously, once they land on the planet, they can be contaminated. But the analysis of this meteorite was very exciting for the astrobiological community. And there were many analyses that were reinterpreted later on. And among them, one of them was the potential presence of biomorphs. If we see this in a SEM image, you may tend to think, oh, that's a worm, or that's a cell that is dividing into different parts. But you have to be cautious because those precipitates can also be formed by processes that are known not to be related to life. I'm just showing here an example of processes that can lead to the precipitation of structures that exactly look like biomorphs. And these ones are experiments that we have run to simulate the conditions of Jezero Crater. And as you can see, you can have beautiful flowers, or you can look at things that look like a spore. So when you see structures that resemble colonies or resemble cells, you have to be mindful that it's not a very strong evidence for life, all of the contrary. However, if you put together different evidences, then you start to have a strong case. And all of this time I've been talking about preservation of the leftovers or the imprint of life. But there is still the question is Mars habitable now? The main reason why we think it's not, at least to our bare eyes, is because Mars is very dry. We don't see liquid water. And all the life forms that we know of on Earth depend on the availability of water. However, with the in situ observations of environmental instruments like REMS and MEDA from the Center of Astrobiology that have been operating in these two landed missions, we have learned that the liquid, although liquid water may not be constantly stable, there is a very intense water cycle on Mars. And the water cycle basically looks like this, that at nighttime, the water vapor that is in the atmosphere, it's absorbed by the regolith. It can condense as frost, and it can also be attached to salts that are present in the regolith and in the rocks and form hydrated salts and brines. And this has been demonstrated based on measurements, and this is one example of the measurements provided by REMS over many years. They have been confirmed later on by Meta, and you can run these kind of experiments in simulated experimental facilities and see how by changing the temperature you actually get the regolith to absorb water and salts to absorb water. So if there is water available in the regolith, then is it good enough for life as we know? Could life use that water and be able to do metabolic activity and reproduction? I'm going to show a graph. It's a bit theoretical, but I'm going to explain the basic idea here. Now, all life forms that we know on Earth, in order to reproduce, need a minimum amount of water, and this amount of water is measured as water activity, and a minimum temperature. If we go below those limits, the cells do not reproduce. And I'm plotting in a graph here the values that we measure on Mars, and in blue is the region that we would call terrestrial-like habitable zone. The measurements from Jezero actually move very far away from there, and the same happens at Gale Crater. So from our terrestrial point of view, the environmental conditions are not good enough for terrestrial cells to reproduce. However, the question remains, could Martian life have been adapted to this kind of water and temperature cycle? The idea behind this is that if eventually we have two planets where independently life emerge or prebiotic life emerge, and then some specific molecules that have very specific functions for life led to the formation of cells and then the evolution of life, those life forms should have adapted on Mars to the desiccation of the planet, to the planet becoming colder and more arid and exposed to radiation. So we have to be open-minded and be ready to look for life as we don't know of. What is a potential strong indicator of life? I was talking about polymers of nucleotides like DNA and RNA. How can they form in a prebiotic environment? There are experiments that have demonstrated that you can polymerize nucleic acids in conditions that simulate the ones of hot springs, for instance. However, when you produce these polymers, they are relatively short and they don't have any information. The difference between these prebiotic molecules and life is the way the nucleotides are organized and the length of the chains. So what we are proposing is to use the detection of these polymers as a strong biomarker. Because as I said, you can have them in prebiotic conditions, which, by the way, would be super exciting if we found them on Mars. But when they become long, then they have information, and then they are strictly associated with life and Darwinian evolution. Furthermore, if you're able to analyze this, you could, in principle, distinguish between terrestrial contamination and Martian indigenous polymers. The way we are proposing to do this is by demonstrating first on Earth how we could handle the analysis of samples. If we have just less than half a gram of a rock, then how we can extract DNA and we can sequence it using nanopore techniques. And we have demonstrated that indeed we could have a rock analog to the ones of Mars. We can cultivate it with only water vapor and see how the microbiome inside the rock grows, and then we can sequence it. And what we observed is that there are microorganisms that we can identify by doing this nanopore sequencing and then doing phylogenetic analysis. And we find microorganisms that are very resistant to radiation, not only UV, but also cosmic radiation, and that they can adapt to survive in very low water content conditions. So the idea would be, why don't we investigate systematically the organisms that survive in rocks, that can thrive in the absence of water, and that can tolerate high radiation levels. By investigating them, then we may be closer to maybe the last potential relative of Martian microorganisms. This would be an example of one of the samples that I would love to analyze. I'm just focusing on this one to illustrate how the analysis would look like. This is one of the samples that Perseverance acquired on the top of the delta. And this is a material that was sedimented with very fine grains. And if you zoom in, you can see that there are some salt veins within the rock that was sampled. Now, these salts are sulfates, and they clearly point to a very late period where there was sulfate or brines rich in sulfates that then evaporated. So the question is, was this environment habitable? And if there was any life, are there any biomarkers preserved in the salts? That is the sample that I would like to analyze. That is the one that I would like to do DNA extraction and sequencing and then compare with all the microorganisms that we know of on Earth. And with this, I would like just to have a postcard to illustrate the collection of samples that have been acquired so far by Perseverance. You can see here some witness tubes. Those are meant to be used as controls for contamination. We can see also the atmospheric sample that was first sealed. And all the rocks that have been sequentially sealed, they represent different stages of the evolution of Mars. So we basically have a time capsule of different moments of the evolution of a planet, which is indeed a unique opportunity to study terrestrial planets in the solar system, and obviously from there maybe to extrapolate to what happens elsewhere in the universe. So that would be it. But before we leave, and maybe for discussion during the question and answer session, I would like to summarize and provide a recipe to search for life. I want you to have a look at this. I encourage you to have a look at this paper where this kind of approach was discussed. And the idea is to provide a framework to report evidence for life beyond Earth, always making sure that we use measurements that are complementary one to another, that they use different instruments, and that they rule out contamination. Among the measurements that we have seen today, we have the study of the mineral composition to make sure that it represents a habitable environment. We would like to look at the total content of organic carbon and then analyze the volatiles and solubles and insolubles organics, analyzing the isotopic ratios of certain elements like the ones we discussed before, and why not looking for more interesting molecules like nucleobases, amino acids, lipids, etc. Another factor that I didn't discuss was the chirality. That's a very interesting biomarker too. And then at larger scale, maybe the formation of colonies or patterns that point to something that has been metabolizing. But to me, the most important biomarker could be to have a biopolymer with information possibilities. And obviously, while you do this, you always have to rule out contamination and any possible process that is not associated with life. So with that, I think I've done a very brief overview of what we know about the presence of biomarkers on Mars based on robotic missions, and I'm happy to answer your questions.